Hey everyone, and welcome. So in this video, we're going to be talking about differentiating Staphylococci and Micrococci. Both Micrococci and Staphylococci belong to the family Micrococci. Micrococci and Staphylococci have some similarities, like both are gram-positive cocci, both are catalase positive, and are usually recovered together, with Micrococci commonly regarded as a contaminant. Now, before talking about differences between micrococci and staph, we'll talk briefly about some characteristics of micrococci. They're gram-positive cocci, usually in pairs, that's 2, tetrads, that's 4, or sometimes irregular clusters. They're catalase positive, just like staphylococci. However, they're coagulase negative, which differentiates them from coagulase positive staphylococci, like S. aureus. They are found as part of the normal skin microbiota and are not considered pathogenic. On culture media, an example like Micrococcus luteus may produce yellow pigments. Now, let's talk about the differences. In terms of mode of respiration, micrococci are strict aerobes. They require oxygen to grow and oxidize carbohydrates like glucose. Staph, on the other hand, are facultative anaerobes, meaning that they don't need oxygen but will utilize it when it's present. In terms of the oxidase test, micrococci are oxidase positive. They possess the cytochrome oxidase enzyme, which is used in the electron transport chain of aerobic bacteria. It's important to note that all bacteria that are oxidase positive are aerobic. Staph, on the other hand, are oxidase negative. Anaerobic acid production from glucose. Micrococci generally do not ferment glucose. They don't produce acid in anaerobic conditions, although there are exceptions like Micrococcus cristinae, which shows weak acid production. Staphylococci generally ferment glucose in the absence of oxygen and will produce acid. This is the basis of the oxidation fermentation test used to separate micrococci and staphylococci. Micrococci will grow on Furoxone twin 80 all red O agar, while Staphylococci will not grow on Furoxone twin 80 oil red O agar. When it comes to differentiating staph and micro using antimicrobial susceptibility testing, generally bacitracin, lysostaphine, and lysozyme are used. Micrococci are susceptible to 0.04 units of bacitracin, resistant to lysostaphine, but also susceptible to lysozyme. While staph, on the other hand, are the complete opposite. Staph are resistant to 0.04 units of bacitracin, susceptible to lysostaphine, but also resistant to lysozyme. So you can use this table to remember. It's actually quite simple. The modified oxidase test, also known as the microdase test, it involves filter paper discs impregnated with tetramethyl paraphenylene diamine dihydrochloride, which is the oxidase reagent, in dimethyl sulfoxide, DMSO. Now, DMSO acts as a permeability agent, meaning it allows the cells to be permeable to the oxidase reagent. In an oxidase-positive organism like Micrococci, the reagent reacts with the oxidase and cytochrome C to form indophenol. Now, indophenol gives a bluish purplish color. As you can see from the picture, the one on the left is oxidase negative and the one on the right is oxidase positive, as you can see from the bluish purplish color. So an organism like Micrococci will give a purplish color, while Staphylococci, which is oxidase negative, wouldn't give a color change or wouldn't have that purplish bluish color.
Alright, here's a question I've next. Can we use the coagulase test to differentiate Staphylococci from Micrococci? Well, generally, no. Suppose we're trying to identify an organism in the lab. We've determined that it is gram-positive cocci and catalase positive. The organism could most likely be Staphylococci, though Micrococci is also a possibility. We will need to carry out more tests to identify the specific organism. If we then carry out the coagulase test, there are two scenarios. A positive result means the organism is a coagulase positive Staphylococci like S. aureus, which is the most common, though there are other coagulase positive staph. A negative result means the organism could be Micrococci, which is coagulase negative, or a coagulase negative staph like S. epidermidis. We will need to carry out more biochemical tests to differentiate Micrococci from coagulase negative Staphylococci, aka cons. Alright, let's do some practice tests. First question, all of these tests differentiate Micrococci from Staphylococci except A. the catalase test, B. lysostaphine, C. anaerobic acid production or fermentation test, or D. the oxidase test. So you can pause the video, think about your answer, and then continue the video. So the correct answer here will be the catalase test. Remember, both Micrococci and Staphylococci are catalase positive. Lysostaphine can be used to differentiate them. The anaerobic acid production or fermentation test can be used to differentiate them. Oxidase test can also be used to differentiate Micrococci and Staphylococci. But a catalase test does not differentiate them because both Micrococci and Staphylococci are catalase positive. Which of the following susceptibility results below is consistent with Staphylococci? So in the order of lysostaphine, bacitracin, and lysozyme. So A. Resistant, susceptible. B. Resistant, susceptible. C. Susceptible, resistant. And D. Susceptible, resistant, susceptible. So you can pause the video and think about your answer. So the answer here will be C. Susceptible, resistant. Remember, Staphylococci is susceptible to lysostaphine, but resistant to 0.04 units of bacitracin, and also resistant to lysozyme. So our answer here will be C. In the modified oxidase test, Micrococcus lutrei will give which color? A. Yellow, B. Purple, C. No color change, or D. Green. So here the correct answer will be purple. So remember, Micrococcus or Micrococci are oxidase positive. So a positive result will be purple or dark blue sometimes. Purple or dark blue. Purplish bluish color. Alright, last question. Micrococci are obligate aerobes. Staphylococci are obligate anaerobes. So A, only the first statement is true. B, only the second statement is true. C, both statements are true. Or D, both statements are false. So our answer here will be A, only the first statement is true. So Micrococci are obligate aerobes, but Staphylococci are facultative anaerobes, meaning they don't need oxygen, but we utilize it when it's present. So Micrococci are actually obligate aerobes, which is correct. However, Staphylococci are facultative anaerobes. Although there are exceptions, but generally that's the answer. So if you've made it this far, I would just like to say thank you. Thank you so much. Please like, share, and subscribe. And if you have any questions related to microbiology, please let me know in the comments below. Again, thank you and have a nice day, guys. Bye-bye.